Okay, let's see this interesting lecture about how cells breathe. By the end of this lecture, you'll know how a mitochondria looks, its structure, its different compartments, and what are they for. Also, you'll know the basics of the production of ATP, which is known as oxidative phosphorylation, and how is this process linked to the process of electronic transportation that creates an electrochemical gradient of protons. You know from the first lecture that mitochondria look like bacteria, and they have their own DNA like bacteria. This symbiotic relation was established a long time ago. We thought that it was between 700 and 1500 million years ago, so to mention a margin, because it's a really big margin. What you don't know yet is that all our mitochondria come from our mothers. And this is because the sperm have their mitochondria at the base of the tail that propels them, and that tail is lost during fertilization. So when the oocyte divides for the first time, it only contains mitochondria from the female. What mitochondria do? What are they for? What's the mission in the cell? Well, the easiest way to remember the mission of the mitochondria is to see them as the power plant of the cell. Let's see. Glucose is the main source of energy. After a meal, glucose is absorbed in the intestines and enters the bloodstream. The resulting hyperglycemia triggers the release of insulin from the pancreas, which binds to receptors in the cell membrane. This makes the cell build and place many glucose transporters in its membrane, so glucose is transported first into the cytoplasm. Now, most of the energy contained in glucose has to be extracted and stored in the form of ATP. So we cannot use the energy in glucose directly. We need to transform it into ATP so the cell can use it to do its functions. You already know one important function of the cell is pumping out sodium that constantly is leaking in to maintain the proper electrochemical gradients across the membrane and also the membrane potential. And this is done thanks to a pump that consumes ATP. Alright, so all the process we were talking about with glucose is called degradation or catabolism of glucose to obtain energy. The reactions involved here are exergonic, and that means that they release energy. So the cell can extract up to 40% of the energy contained in a given molecule like glucose. And just in case you can see that this is not much, think that the most efficient car engines can extract a maximum of only 25% of the energy contained in fuel. So the cell wins because the nature's design is much more efficient. Okay, let's retake the story we were with. So we have the glucose, it's already in the cytoplasm, and what's going to happen next? Well, first glucose enters the glycolysis circuit. If you don't know, glucose has 6 carbons, and after glycolysis, 2 molecules of pyruvate with 3 carbons each are produced. Here, some chemical bonds have been broken and a small quantity of energy has been released, but most of the energy still remains in the pyruvate molecules. This remaining energy will be released by the mitochondria, but only if there is oxygen available, and this is the reason why we breathe. If there is not enough oxygen available, for instance, when you make a great effort, pyruvate stays in the cytoplasm and can be further degraded, although in a much less efficient manner that we know as the lactic fermentation. And the end product is lactate that still contains energy. So some energy is not extracted and used. You probably experience the feeling of lactate when it accumulates in your muscles. Is that feeling of having to stop when you exercise for too long. That tiredness when running a long race or climbing a mountain. In fact, training is the way to optimize the removal of lactic acid from your muscles so you can perform better and better as you go on training. Alright, this formula here shows that a molecule of glucose can produce 2 molecules of pyruvate, 2 ATPs and 2 NADHs. These NADHs can be converted later to 3 ATPs each. So, if only glycolysis occurs, we can extract only 8 ATPs in total, and this is not too much for a so energetic molecule. All right, by the end of this presentation, you'll see that we can extract up to 38 ATPs if we can degrade the glucose molecule completely. Okay, let's state first that you'll see pyruvate or pyruvic acid. All right, these are exactly the same thing, so you don't get confused. All right, so why do we breathe then? Well, respiration allows the total degradation of pyruvate to render carbon dioxide and water. And this includes 2 processes. First, we have the cyclic acid cycle, most commonly known as Krebs cycle, and later the electron transport chain that is matched to oxidative phosphorylation. But to understand these 2 processes, first we need to know the particular structure of a mitochondria. This organelle has 2 membranes, an external one and an internal one. The inner membrane is much larger, so it forms folds like, we call them crista, and you can see them in this image where the external membrane is cut off. The space between the membranes is known, this one is a very important space, and is known as the intermembrane space, and the space inside the inner membrane is known as the matrix. The matrix is very rich in coenzymes, phosphates, and other molecules needed for respiration to correct produce ATP. Also remember that the outer membrane is permeable to small molecules, but the inner membrane is only permeable to pyruvate, so it can go in the membrane, and ATP, because ATP will be produced inside, so you need ATP to go outside so it can be used by other processes. All right, let's review now the electron transport chain, which is the first step in the production of ATP. The inner membrane is 80% proteins because it contains the enzymes of the electron transport chain, while the Krebs cycle enzymes are located inside the matrix, floating there in that solution. All right. So remember that mitochondria is going to produce 95% of the total ATP produced by a cell, and this is what it makes them essential. All right. So as you see in this image, the initial catabolism of glucose is initiated in the cytosol and produces pyruvate that enters the matrix to be converted into acetyl coenzyme A. And this product is the one that can enter the Krebs cycle. Not here in the right of the image on top, note that fatty acids are also can be converted into acetyl coenzyme A through a process that we know as beta oxidation. All right, so if you look again in the left for glucolysis, you'll see that NADH is produced, and this one has to enter to the matrix where it will join with the NADH and FADH2 produced by the Krebs cycle. All right, so and you'll see that these products have to be in the matrix to be used. The Krebs cycle, as you see here, also produces carbon dioxide. That is what you breathe out with every exhalation. Okay, NADH and FADH still contain much energy, and that will be used to pump protons into the intermembrane space. All right. Okay, before continuing, let's say that for every molecule of NADH will render at the end 3 ATP molecules, and every molecule of FADH will produce 2 ATPs, and this is done by means of successive redox reactions by proteins of the electron transport chain that are located in the inner membrane of the mitochondria. Remember that the hydrogen atom has 1 electron and 1 proton. You see it in that video running. Okay, so the one spinning around is the electron, and in the nucleus we have the proton and the neutron. The neutron doesn't have any charge. All right, so the 4 protein complexes of the electron transport chain that you see in the lower image, these are named 1, 2, 3, and 4, can use the energy of electrons to pump protons, which are represented by the sign H+, in the intermembrane space. This is going to create an electrochemical gradient of protons between that space and the matrix. So now protons are pushing hard to move into the matrix. The final electron acceptor, as you see here, is complex 4, and this one is special because it can combine 2 electrons, 2 protons, and oxygen to form H2O, which is water. So protons that have been pumped in the intermembrane space can re-enter the matrix only through a special protein that is called, the complete name is F1F0 ATP synthase, that uses the electrochemical gradient energy to create the chemical link between ADP and phosphate to form ATP. This protein, well, you don't see it in this drawing, okay? So let's go with it. Okay, let's see now how ADP is phosphorylated to ATP, which is the final objective of all these processes. how this happens is based in the what we call the chemiosmotic theory. It was proposed by Mitchell in 1961, and he won later on the Nobel Prize for his discovery or his proposal. Okay, so what he proposed is that the electron transport and the oxidative phosphorylation to ATP are coupled processes to the proton gradient we just explained a minute ago. All right, so the huge electrochemical gradient energy created at both sides of the internal mitochondrial membrane is used for the phosphorylation of ADP to ATP by the protons being forced. Well, they want to go through the membrane, but they are forced to pass through this special ATP synthase, which is a protein of the turbine type. So the protons, when they go through it, as you see, it turns, and one of the units turns, and the other one is acting as an enzyme and producing the ATP. Okay, so F1, F0 ATP synthase consists of two protein complexes, which we name F1 and F0. F1 is located in the matrix, consists in 9 subunits attached to F0. This is the enzymatic portion of the ATP synthase that catalyzes the binding of ATP and inorganic phosphorus and forms ATP. F0 represents the portion of the ATP synthase that crosses the inner mitochondrial membrane and forms a tunnel to permit the passage of protons to the matrix, pushed by their electrochemical gradient. It's important to remember that electrons will only flow through the electron transport chain when all substrates needed for the synthesis of ATP are available. So you need to have oxygen, all right? You need to have ADP, and you need to have inorganic phosphorus, okay? All right, let's see now an animation of all these processes. So you don't have to use that much your imagination.
All eukaryotic cells, from yeast to those that make up the human body, contain membrane-bound organelles with specialized functions. Mitochondria are double-membraned organelles that harness most of the energy that cells need to grow and reproduce. Nearly all of this energy comes from reactions that take place at the inner mitochondrial membrane. One of the key roles of this membrane is to act as a barrier to positively charged particles called protons, thus allowing a concentration gradient to be maintained where the intermembrane space has far more protons than the matrix. The membrane also contains a large protein complex called the F1-F0 ATP synthase, which uses the proton gradient to drive the synthesis of ATP molecules. These ATP molecules ultimately provide the energy for most of the cell's reactions. Just as man-made power plants produce electrical energy by using the flow of wind, water, or steam to rotate a turbine, the synthase makes ATP by using proton flow from one side of the inner membrane to the other to rotate protein subunits. If there is no proton gradient, synthase subunits stop rotating, and the cell can quickly become starved of energy and die. Therefore, the protein complexes and small molecules that establish this gradient and maintain it play an essential role in the life of the cell. At the heart of this system are 4 protein complexes, numbered 1 through 4. Complexes 1, 3, and 4 directly pump protons from the matrix into the intermembrane space. Complex 2 does not directly pump protons, but it does promote proton pumping in complexes 3 and 4. Proton pumping requires energy, and the 4 protein complexes get this energy by transferring electrons through a series of coupled reactions. This linked process of electron transport is why the 4 complexes are collectively referred to as the electron transport chain. Let's focus on complex 1. A byproduct of sugar metabolism called NADH deposits 2 high-energy electrons in complex 1, where they are passed along a chain of redox centers. Redox centers are clusters of atoms that have different affinities for electrons based on their unique atomic configurations. Let's closely consider a pair of redox centers to reveal 2 reasons why an electron moves from the top redox center to the bottom. First, the bottom redox center has higher affinity than the top one. Second, the distance between these adjacent redox centers is ideal for an electron jump to occur, which explains why electrons typically don't bypass the bottom redox center. A small amount of energy is released each time an electron is passed between redox centers. Complex 1 harnesses this energy across all the redox centers and uses it to pump protons. The last redox center in complex 1 donates 2 electrons to a coenzyme Q molecule. Complex 2 is similar to complex 1 in 2 important ways. First, high-energy electrons also enter complex 2 via a byproduct of sugar metabolism, although here the molecule is FADH2. Second, complex 2 also transfers electrons between several redox centers before donating them to coenzyme Q. One major difference, however, is that complex 2 does not use the energy liberated to pump protons. Coenzyme Q molecules from complexes 1 and 2 donate their electrons to complex 3. One electron is recyclable and can re-enter complex 3 later, but the other passes through 2 redox centers before reaching cytochrome C. Cytochrome C carries the electron to complex 4. The electron transport chain ends in complex 4, where a series of reactions involving 4 electrons converts a molecule of oxygen to 2 molecules of water. The proton gradient is strengthened because 4 protons from the matrix are incorporated into water molecules, and another 4 are pumped into the intermembrane space. In the absence of oxygen, the electron transfer comes to a halt, meaning that ATP synthesis also stops. Indeed, the reason we breathe oxygen is so that it can serve as the final electron acceptor at the end of the electron transport chain. In this animation, we have explored each protein complex in isolation, but in reality they are very densely packed. Together, they effectively make the entire surface of the inner mitochondrial membrane a giant cellular power plant.
Alright, so finally here you have a table showing the neat result in ATPs of a single glucose molecule from the time it enters the cell cytoplasm until its total degradation to water and carbon dioxide. Depending on the cell type, the final result may be 36 or 38 ATPs, because some ATPs are sometimes spent in a step to transport the two NADHs of glucolysis into the matrix. All right, so it is variable, but you see that the difference of the total degradation compare with only glycolysis is very big. All right, so I hope it was clear enough. I'll see you in the next lecture. Thank you for listening.